This Week in Virology, the podcast about viruses, the kind that make you sick. From MicroTV, this is TWiV, This Week in Virology, a special episode recorded on October 17th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Today we are back in Atlanta, Georgia. We're at Georgia State University in the Institute for Biomedical Sciences. And I have 4 faculty here. We're going to talk with them about their research. And joining me today on my right, Chris Basler. Welcome to TWiV.
Thanks.
And next to Chris, Priya Luthra. Welcome.
Thank you.
And you're the reason I'm here.
Yes, I'm glad you could make it here.
So you can either blame her or thank her, I guess.
We'll see how it goes.
That's right. On my left, Richard Plemper. Welcome.
Thank you.
And Andrew Gewirtz.
Thank you.
You're welcome. I want to start by just exploring your histories a bit, where you're from and where you trained. Chris, let's start with you. Where are you from originally?
I was born in New York City.
Wow.
I grew up in northern New Jersey,
Which
Morris
town?
County, Rockaway Township.
So I grew up in northern New Jersey also, in Bergen County.
Okay. Well,
Close.
A little bit. Yeah, not too far away.
Where were you? What city were you born in?
Brooklyn. I was in...
Oh, you were born in New York
Both
City.
my parents are from New York City, yes.
I didn't know that. Did you know that?
No.
That doesn't come up that often during the daily
Right.
course of work.
It doesn't. Did you go to high school in New York City?
No, I was in New Jersey from the age of 1 to 18.
Okay.
So,
Then college was?
Lemoyne College, a small school in Syracuse, New York. And from there I went to grad school at Albert Einstein College of Medicine, where I trained with Marshall Horowitz, looking at adenoviruses in the E3 region for immune modulatory functions. And was there for about 5 years, and then moved on to Mount Sinai, where I trained with Peter Palese. Not planning to stay there quite as long as I did, but I wound up starting as a postdoc and going to full professor. So I was at Mount Sinai for about 20 years.
So we have several things in common. We have New Jersey and Peter Palese.
Yes.
But we did not overlap. I left in 79.
And I arrived in 1995.
Yeah, quite a bit later. So do you remember when you got interested in science? How far back?
I think I was interested in science from very early age.
Yeah.
And I'm not sure exactly why that was. It just caught my interest.
Your parents weren't scientists?
No, my mother was at the time a housekeeper, and my father's a banker.
A banker, it's good. Maybe that helps is raising money for science, right?
Wish it did. So far, that hasn't really worked that way.
So I knew Marshall Horowitz quite well. He was a really interesting and nice guy and really important adenovirologist, so it's a good person to train with. And Einstein was at one time a mecca for virology.
Yes.
I think there's still quite a few virologists there.
I think
We
there
have
are.
Kartik Chandran, we have Margaret Keelian,
Right? I guess.
and others.
Yeah, John Lai is a biochemist,
Right.
structural biologist who does a lot of antibody virus work,
Okay.
some HIV people.
Priya, you're from India, I know
Yes.
that. Which part?
Delhi, it's north.
Oh.
And
When did you come here?
back in 2005, and I think that was my first trip that I actually sat in a plane and took my flight to US.
2005.
Yes.
So you had gone to college by that point.
Yeah, so I did my undergrad. I was trained as an engineer. I did biotechnology. I think if I think back about the science, I was always interested in science, but back in the day I was in an engineering school, so I didn't have much experience doing actual research. But during the last semester, we are supposed to do a project, like a research project.
Right.
And that time I got this opportunity to work in Indian Institute of Immunology, which is one of like the big ones. And I think I kind of looked, that was the first time I touched tissue culture. And then I'm like, okay, I should apply to grad schools in US. And I applied and I got in Penn State. came here in 2005. And then Penn State was an interesting program because it was integrated biosciences. So I didn't have to choose a discipline before I came in. So I rotated in different labs from C. elegans to Drosophila and immunology. And I think I stuck with virology. That was the first time I was in Viauhe's group. And he was at that time doing experiments himself, and he was running plaque assays and stuff. I'm like, oh, this is interesting. And it just stuck with me. And then I'm like, okay, this is what I want to do. And since then, I've been thinking, he was the one who introduced me to virology. Before that, I've never thought of viruses, never thought of being in this field, but it's been good so far.
So after that, did you do a postdoc?
So yes, so that's when, at the end of my PhD, Biao got a position at UGA.
Right.
So we moved to
Right.
Athens. So I was in Athens for a year with him and finished my PhD at Penn State. And then I was applying for postdocs. And then I think at ASV, I contacted Chris that I'll be presenting. I was looking for postdocs. And at that time, my husband was in New York. So New York was like a obvious choice for me to find like a postdoc. And then Chris had a position, and it worked out. And I came to Mount Sinai, and since then I've been with Chris, starting as a postdoc, changed to instructor, and then he got a position at Georgia State. So I followed his footsteps, and I came here. Yep.
Right. So Biao was a trainee of Bob Lamb,
Yes.
and Bob Lamb was on my thesis committee.
Oh, okay. Good connection there.
Just threads that are in science all the time.
Yeah.
And I visited UGA, and Biao was one of my guests.
Yeah, Adrienne, my former student, I think she's the
Yes.
one who invited you.
Right.
Yes.
She invited me at, it was a ASV at Fort Collins, I think.
Yeah.
And then, then she invited me to UGA. And I was telling people yesterday, for breakfast, we went to a little diner because she wanted me to have grits. She wanted me to have a Southern experience.
Yep.
But if you've seen this movie, My Cousin Vinny,
Oh, yeah.
anybody seen that?
Yes.
Yes.
So I have an affinity for it because it's me. But grits plays a role in that movie, which is too long to talk about, but pretty cool.
So did you have the regular or the instant?
I didn't have any grit. Oh, at the UGA, I had regular. They cooked
Regular,
them, yeah,
uh-huh.
which take a while to cook.
That's what I've heard.
Yeah, and that's part of the thing about the movie. Yeah, it's cool. So Andrew, sorry, where are you from?
So I'm also from the New York area, born in the Bronx, but really raised in Westchester County. Went to State University of New York in Albany as an undergraduate. Was a physics major. And then I ended up working as a lab technician and getting a PhD in biochemistry. Did a short postdoc in the Boston area, and then moved to Atlanta. I was at Emory for about 15 years or so, and focused on intestinal biology and the gut microbiota. And from that, started to think about how viruses might interact with that, particularly in terms of rotavirus. So that's really what kind of brings me to this this podcast, I guess, and our work on rotavirus.
So you told me at the beginning you're not a virologist, but you've published a cool paper involving a virus. So
Yeah, I will take credit for that. Sounds about right.
So the person in my lab, Amy, sent me your paper last week. She said, you should have this guy on TWiV. And then Priya sent me the paper. She said, you might want to have Andrew on. So
Okay, all right. I had assumed the effort was coordinated. I didn't realize.
it was pretty good timing. And so, there's someone at Columbia who works on SFB. So that's also why it got my attention, but we'll talk about that.
Okay.
But we can tell from your accent that you're from New York.
Really?
Well, I can. It's clear. Right.
All right.
Nobody else gets that? No?
I mean, I guess
You should, Chris.
If
Yeah.
you had to ask, I guess that would be my guess.
Yeah.
I would profess to have the Brady Bunch accent, having grown up watching a lot of TV. But if you hear it,
I do hear it, yeah. Do you know Nate Landau?
I do not.
So he's a HIV guy currently at NYU. And if you listen to him, he's been on TWiV. It's you.
Okay.
Exactly. And he's from New York as well. Richard, where are you from?
Well, I'm originally from northwest Germany, so just about 50 miles north of the old heavy coal and steel area, almost Netherlands. And so I grew up there, and then decided to go south to southern Germany for studying. Part of because the weather is much better there, and then partially at the time in Germany I wanted to study biotechnology, and so I actually thought I have a career in bio industry. And at the time there were only 2 places in Germany where that could be studied, and so Stuttgart in southwest Germany was one of them. And so I joined what was at the time just a brand new biotechnology course that they had, or program that they had launched. And then while there, I realized at some point during what was a diploma at the time in Germany that I really don't want to spend all my life steering in very large pots, and rather want to go more the smaller molecular biology route. And so I then did my PhD there in working with yeast and on secretory system and protein folding, protein quality control. And that really got me, after the PhD, into virology, because a lot of, obviously, what we know about secretory system, virus and viral proteins were really the model systems that have revealed many of the very fundamental cell biology aspects that we now take for granted in textbooks.
Like Ari Helenius's work.
Exactly. Yeah, he was the big role model. And so that got me into viruses. And then when I finished my PhD, that was in 1999. So then I came from my first postdoc to the US, and I was to Mayo Clinics, and they had just started a molecular medicine program wanting to cure cancer with cancer viral therapy, which of course we know is now very big field and was at the time, but still I think not quite as broad as what has unfolded since. And so I joined the lab of Roberto Cattaneo, and working then on paramyxoviruses, on measles virus, and stayed there for a couple of years, and then really got very much hooked, less on the therapeutic aspect, but more on the basic virology, in particular at the time of viral entry, membrane fusion. As you know, paramyxoviruses are some of the experts when it comes to fusion activity. And so I then went to a lab that was very well known, in particular in this area, and that was Dick Compens here at Emory. And so I joined a couple of years later. I joined Dick's lab, first as a postdoc, and then as a research professor. And then, as you know, Dick is very supportive of his trainees, and so I got my first major grant in still while being in Dick's lab, and then from there moved to pediatrics at Emory, where I had my first faculty position.
So some of these people have been on TWiV. Roberto has been on TWiV.
I know, yeah.
Yeah. And recently he sent us his paper and asked us to discuss it, actually. This one showing trans movement of proteins from cell to cell.
The close biology, I believe.
Yeah. Which he said
very nice.
He really was excited about. So as TWiV gets older and older, more and more people who are mentioned we have talked with. Peter, of course, has been, maybe not so many in your, because you don't.
Have you had Mary Estes on from Baylor?
Oh, Mary Estes.
That's who really got me into this area.
Yeah, I've known her for years, for sure. I don't think so. I visited Baylor once, and I had Richard Lloyd and the former chair. What was her name? She worked on SV40. Incredibly important virologist. Oh my gosh, who am I thinking of? SV40, she showed that transformation of cells is mediated by T antigen. She had made a temperature-sensitive mutant of T antigen, which you could turn on and off by heat, and the cells would be either normal or transformed, depending on. And she was the chair for many years. I have to look this up. Baylor, SV40. Let's try that. Gosh, because I can't not. Janet Butel. You know the name. Very important person in the field. Anyway, she was on. But Mary Estes, we need to get on. Yeah. She's a little shy.
Not once you get to know her.
No, but she's about being on the podcast.
Oh,
It's funny. She said, I don't know what I would talk about, which is a funny thing to say. Okay, so let's talk a little bit about your science. Let's start with you, Chris. You've done a lot of work on Ebola viruses. So did that emerge from some of your previous training? You said adenovirus with...
Yeah. Well, I mean, so I got into the Ebola field sort of by accident as a side product of things we were doing on influenza virus in Peter's lab.
Peter's lab, okay.
So I was still a postdoc at the time. And, at the time, we were, I guess, a few things had happened. So plasmid-based reverse genetic systems for influenza virus had just started to work, both in Peter's group and Yoshi Kawaoka's group. And I guess my contribution there simply was I got it working a little bit better, higher efficiency. And we were also collaborating with Jeff Taubenberger, Adolfo Garcia Sastre on 1918 flu. And we're starting to do the first, make the first recombinant viruses that contained genes from the 1918 virus, which is work we actually did at the Southeast Poultry Research Lab here in Athens, Georgia, because it was an available high containment lab for the time. And one of the initial hypotheses was that the NS1 protein of influenza virus, which is a suppressor of interferon responses, maybe that was better in the 1918 virus, and that was contributing to high virulence. And so I became interested in looking for ways to screen for relative capacity of different viral proteins to block interferon response. And so we developed an assay that relied on an influenza virus that lacked the NS1 protein. And we were simply transfecting plasmids, trying to complement growth of the virus in cells that could make interferon. And so the thought came that we could use this assay to look at different NS1 proteins from influenza virus, but we could also take the same assay and start screening other viruses for proteins that would block interferon response. And at the time, so this was, I guess, around 2000, Ebola virus was kind of a, I guess from my perspective, a cool sounding virus, right? It certainly was noted for being highly deadly and scary. And I think there was some concern of it as a potential bioweapon at the time. But we published our first paper showing that the VP35 protein from Ebola virus was a suppressor of interferon response. And the first assay was it could rescue growth of this mutant influenza virus.
Okay.
And so that was a paper. We published it in PNS in 2000. In 2001, obviously, bioterrorism and how one responds to bioterrorism became, a big deal.
Yeah.
And Ebola was high on that list. And so, that was really my path to funding, which was my path to tenure track faculty position.
Right.
So it's sort of a accidental offspring of the influenza work.
It's a good example of how science works, right?
No, absolutely.
Always. You just find things without really knowing where they're going to lead. And
Right.
so that you started with VP35, and then there are two other suppressors encoded in the Ebola genome, right?
Right. So, we've looked in both, there's Ebola is a filovirus. So within the filovirus family, there's, I guess now multiple genera, but the two big ones that have caused disease in people are the Ebola virus genus and Marburg virus genus.
Right.
And they cause similar disease, but the viruses are different, right? Most of the protein amino acid identity is around 30% between the two viruses. So we've looked both in terms of how does Ebola and Marburg block interferon responses. And so Marburg also makes a VP35 protein that does much of the same thing that the Ebola virus.
So what does the protein do to block interferon?
So it's blocking the RIG-I pathway, and this is a work that Priya has done a lot on. But it's a double-strand RNA binding protein. So one model is that it binds and sequesters the double-strand RNA that can activate RIG-I-like receptors. But Priya has also shown that it interacts directly with a protein known as PACT. And PACT is a protein that facilitates activation of RIG-I and MDA5, the other cytoplasmic RNA sensor. And so VP35 can block packed rig-eye interaction as well. So we think that also is contributing to the suppression.
So both Ebola and Marburg
Marburg
VP35 have that same activity.
Yes. I mean, they're very, very similar. But the question was, and there was some data in the literature to suggest that if you infected cells with Ebola virus and then treated with interferon, that the ability of cells to respond to the exogenous interferon was blocked as well. And that's a function that VP35 didn't seem to impair. So we looked for proteins, and all along, the assumption for all these assays was that we could find a single protein that would be sufficient to carry out these activities. And we were lucky that that proved to be the case. And so for Ebola virus, we found that the VP24 protein, which is a small protein involved in virus assembly, was also a suppressor of interferon signaling. And there, the data shows that if you put VP24 in cells, or if you infect cells with Ebola, and if you treat with interferon, you normally see tyrosine phosphorylation of STAT1 and STAT2, and then they move to the nucleus within about 30 minutes. And VP24 doesn't block the phosphorylation events, as far as we can tell, but it blocks nuclear translocation of phosphorylated STAT1. And that correlates with its ability to interact with the same important alpha proteins that normally would translocate STAT through the nuclear pore. And so we can show that they can compete for binding to the important alpha proteins.
So 24 will prevent cells from responding to interferon, exogenous interferon.
Yes, right.
Okay, and so it's different from 35, which is blocking the sensing part.
Right, and blocking the initial induction of the interferon response. And then, so we also looked in Marburg virus, and it turned out the VP24 protein for Marburg virus didn't block the interferon response. And if you look in Marburg virus infected cells and treat them with interferon, the picture is different because now you block all the phosphorylation events you typically see. So you don't see phosphorylation of STATs, you don't see the phosphorylation of the JAK tyrosine kinases that carry out all the phosphorylation events. And then we screened Marburg virus proteins for the ability to block these responses. And the matrix protein BP40 is blocking that activity.
And is that also the case for Ebola viruses?
So no, not as far as we can tell, that the VP40 does not have any significant ability to suppress interferon signaling.
So for Ebola viruses, VP35 and 24 are they're suppressing different parts of the interferon pathway. And for Marburg 35 also is a suppressor, but then in addition, VP40 of Marburg is what did you say it did again?
So it's blocking JAK1 activity as far as we can tell.
And so what strikes me is that there are multiple antagonists, at least in Ebola viruses, right? Which seems like overkill for a virus with how many proteins? 9 or?
Well, it depends how you count. But 7 transcriptional units, and in Ebola, the glycoprotein gene gets edited, so there are probably 9 main translation products. Right.
So you must think about the reasons for having multiple antagonists, right?
Right. And I mean, I think clearly there's rationale for wanting to block both components of the pathway. But if the virus infects a cell and you immediately turn on interferon response, that would seem to shut the virus down right away. And that's what we see in cell culture. And we can with collaborators have made VP35 mutant viruses that are unable to block the interferon response. If you put them into cells that can make interferon, that mutation takes the cell from essentially no interferon response to a very, very robust interferon response. And we've actually recently published a paper, and Priya was involved in this as well, that we've now taken this into non-human primate models, which are the preferred model for Ebola pathogenesis. And these viruses really are severely impaired. So we can put 300,000 infectious units intramuscularly into animals. They show very slight viremia. A couple of the animals showed a slight fever, but they recover, and that mutant virus behaves like a live attenuated vaccine. They're protected from wild-type challenge. So clearly that's important.
And so normally a wild-type would cause hemorrhagic fever in this?
Yes. These animals die usually a little bit more than a week, very uniformly, the severe hemorrhagic
By the way,
disease.
I noticed you did this in Galveston,
yes,
where I was 2 weeks ago,
very good.
visiting GNL, and put on a suit and all that stuff.
Yeah.
And I spoke with Chad Meyer, who I think was on one of the papers.
Yes. Yeah, we've published a few papers with Chad and with Tom Geisbert.
So that's because you need a facility that can not only deal with the virus, but non-human primates. That's what they have at Galveston.
Right. Yes, and there are very few of those in the world, and they do it well there.
So the mutations that inactivate VP35,
right.
so that they don't interfere with whatever, it has another function for the virus? Or?
So it has at least two other critical functions. One, it's an essential cofactor for the viral polymerase.
Okay.
So if you don't have VP35, you don't have any viral genome replication or gene expression.
Okay.
But so we can, and it also plays roles in assembly of the virus. It's a structural component of the virus. So we can build point mutations into the RNA binding domain that disrupt double-strand RNA binding activity, largely disrupt the ability to block interferon response, but the replication functions are essentially intact.
Okay.
So otherwise those are relatively robust viruses.
And are there multiple, I guess what I'm getting at is does it ever revert? Is it a stable? So polio vaccines are not good because they're just a few mutations. They revert all the time.
Right.
Would this be more stable genetically?
So, part of the reason I don't have a definitive answer is just the BSL-4 work is always so cumbersome that it's difficult to do there what would be straightforward in BSL-2. So we haven't really rigorously looked at potential reversion in cell culture.
Yeah.
In the animals, the monkeys, the virus barely grows, so we don't really have significant material which we can sequence to see what happens in vivo. So it's obviously an important question and something of interest. We just don't have a clear answer.
Yeah. So I got that all the time in Galveston, how hard it was to do basic experiments. It takes too long. You can't compete in grant terms. It takes too long. And they were really, it was really an interesting approach, which I had visited the BSL-4 in Boston, the needle, which hadn't been open yet, so they didn't get to that point, but it is hard. But you're not thinking of making a vaccine out of this, right? Because there are...
No, this is more proof of principle that VP35 is an important virulence determinant.
Sure. Now, the other thing I wanted to ask you about, for Marburg, it's clear, I think, that the natural reservoir is bats, right?
Yes.
Still not clear for Ebola virus. Would you agree with that, or?
Yeah, it's still, I don't think anyone's isolated a live Ebola virus from a bat. There's lots of circumstantial evidence and isolation of what's apparently viral Ebola-like RNA from bats, but it's not definitive at this point.
Yeah, I asked Tom Kizak that in Galveston, and he said it's not enough. And he said it's really hard to do.
Yeah.
So it's not clear that we'll ever get there. But anyway, these immune suppressors, do they have similar functions in bats or bat cells?
Yeah, so that we've done, and that's a set of experiments we did in collaboration with Alex Bukriev, who's also at UTMB.
He's at Galveston, yep.
And so Alex made Ebola viruses with mutated VP35 and also mutated VP24. And we were involved in a study where we're doing RNA-seq analysis of human cells versus bat cells infected with these viruses and also infected with Marburg. And you see a very similar pattern that these viruses don't trigger an interferon response. The wild-type viruses don't trigger an interferon response, but you mutate VP35 and suddenly you have a very robust
bats do make interferon, and so in order for these viruses to reproduce in them, they have to antagonize it, apparently.
That's where the data points, yes.
But isn't it true that in bats the outcome may be different? It doesn't always make bats sick?
Yeah, so experimental infections with Marburg virus in bats really has, the virus will replicate some. I think if you look in organs, you see some evidence of inflammation in the liver, for example. But otherwise, the animals are fine. There's evidence that they can shed virus.
Yeah.
And that's probably how the virus maintains itself in nature. People put Ebola into the same bats that are known to be reservoirs for Marburg, and the virus barely grows. So the thought is that that's probably not the right bat as the reservoir for Ebola.
So if you take out this immune, so say VP35, then you probably convert it into a lethal infection in bats or bat cells, right?
No, I would expect just with the same as with a human or non-human primate, that if you don't have VP35, the interferon will turn on, the virus will get shut down, and it won't cause disease.
It's the opposite. So what I'm trying to understand is somehow in bats, the virus has evolved to not kill them like it does humans.
Right.
Because it's been in bats for years and it's new every time it comes into humans. So maybe these immune suppressors are overkill in humans, and that's why they're so pathogenic to begin with.
Yeah. I mean, that's one hypothesis.
Yeah.
I mean, I guess if you profile what's going on in an infected cell and cell culture, they look the same, that you don't see a response really in either system. So it's hard to say at this point that can explain the outcome of infection.
Now, so I think you believe that the huge response to infection is really the key to the hemorrhagic fever, right?
Sure, right.
The production
so it's sort of there's a dichotomy, I guess, between what you see in cell culture and what you see in a human or a non-human primate. That the virus that completely suppresses an interferon antiviral response in cell culture, in vivo after 3 or 4 days in a monkey is, causing this overwhelming inflammatory response, and which is accompanied by high levels of interferon-induced gene expression in lots of different cells, and ultimately that leads to death in most cases.
So the other thing I wanted to ask you about is Loviu virus, which was identified in a cave in Spain originally. Is that correct?
Right. So there was a die-off of bats in a cave in Spain.
Right.
And virus wasn't isolated, but near full-length viral genomes that looked very much like an Ebola virus or a Marburg virus were isolated. And then there's a similar case. There's a virus, I'll mispronounce it, but it's Mengele-virus, which was recently described as, this again is a virus which hasn't been cultured, but is another filovirus, and where they found RNA in the liver of a bat, and it's a near full-length genome.
Right.
So it's a theme that there are filoviruses that we haven't experienced and haven't been known to cause disease in people, that you can find in bats, various places in the world. And
you showed that the 24 and 35 are also immune antagonists from Loviu, right?
Yes, that's right. So, and they're behaving very much, at least in our transfection assays, as you would expect for an Ebola virus protein. The Mengele is interesting because we've done similar experiments with that virus now, and there, phylogenetically, it looks a little bit more like Marburg than Ebola. And when we look at which protein blocks the interferon signaling pathway, now it's not the VP24 protein as it was in Yoviu, but it's the VP40 protein, which is behaving a lot like the Marburg VP40 protein.
So I think that's interesting, where you don't have infectious virus, but you can take the proteins and ascribe some functional activity to them.
Right.
And get an idea about the evolution and so forth. But we think the host of Loviu is a bat somewhere, or bats. Right?
Presumably, because that's where the RNA was found.
Yeah. Okay. So, Priya, you've participated in a lot of this over the years, right?
Yes, since I've been with Chris.
So what would you say your overall interest is? Are you interested in these immune suppressors?
Well, so I think when I came to Chris's lab, my main interest was looking more into innate immune responses and how viruses tackle those. Yeah. And that was a very good starting point for me, because at the time when I came to the lab, there was this very interesting paper describing on the RIG-I signaling. This PACT came along, and it's like an activator of RIG-I pathway. And then Chris, when we had our first meeting, Chris showed me 2 papers. This was one of the PACT paper, and there was another one. I can't remember at the point what it was, but it was also something to do with innate immune response. But in general, the lab interest was how the viral interferon antagonists are working and how they're antagonizing these immune responses. So I was like, I'll take a shot at it because I was interested in that pathway, and that was my first finding with like the PACT and RIG-I and BP35. And that was actually pretty cool because I was interested to begin with in those pathways and figuring out how the viruses are like targeting that. And that was my introduction to BP35. And since then, I've been interested in general virus host interactions. A lot of work has been gone in those lines, finding the host interactor that can either curtail the BP35 function either in the interferon antagonism or the virus replication. And over the years, also thought came down, can we target these functions of the virus itself? Can we generate like a high throughput screening assays? And that's a lot of, I think last couple of years have been like the effort in developing high throughput screening assays.
To find inhibitors of
For
like
BP35.
35?
Yeah.
Okay.
BP35 and the replication of the virus, because we have this very well standardized mini genome system, which is like a mini replicon, and we can study the virus transcription replication in by using transfection of the plasmids.
So
and
what would be the readout for a VP35
so
inhibitor?
so like I said, so the first assay is like using an interferon reporter. So converting that, because we know if you have a double-stranded RNA-induced interferon beta production, if you put VP35, it inhibits that signal. So our idea was to find like a gain of function approach where we were looking for small molecules that would prevent VP35 from inhibiting that response. Okay. So interestingly, I mean, I think the screening assay was really good. We had like 384 assay. We did a lot of screening. I did my self-screening going to Harvard, the screening core, Microbiotics, which was also our collaborator in this. But at the end, we end up finding a lot of DNA damaging compounds, the stuff that was just, causing a lot of DNA damage, triggering a type 1 interferon. But the interesting part that we really found was that BP35 itself lost this ability to block that interferon response, even though it was generating a type 1 interferon response. And it was not like a direct blocker of BP35 itself. So overall, we didn't end up finding something that was directly targeting this viral protein, but we end up identifying a lot of mechanism that was not targeted by the virus, especially VP35, and that end up merging of the DNA damage response pathways and cGAS and STING pathways. So the type 1 interferon that is triggered by these two specific pathways, we found that a lot of interferon antagonists, including VP35, do not block the type 1 response. So that's like another way of saying that if you have like a small molecule that can induce a response, not just putting cells with like treating with interferon, because that can change a lot of things.
So basically the compounds damage DNA and that activates the cGAS pathway.
Yes. And that...
Okay, which is not what Ebola virus normally does.
Right.
No.
Although it could, because dengue does apparently, right?
So the dengue, I was involved in that work with the Anna's lab.
Fernandez, Yeah,
I think that's how the merging of the two concept collided. I'm like, it's worth testing it because a lot of this stuff is done in 293Ts. Cells and 293Ts do not encode cGAS or STING, right? You cannot, in the basal level, you don't see a lot of expression.
That's why we use them for DNA transfection, right?
Yes.
And they're also lacking something else.
There are a lot of things that's lacking. Just TLR.
TLR, yeah, but the cGAS is an important part of it. But also, if you add inhibitors of histone deacetylases, you can even get better transfection because the DNAs get in the nucleus and they're chromatinized and so forth. Yeah, I love how we figure out these old cell lines that we used years ago. Probably you used 293s in Marshall's lab, right?
I don't think we did. I think I only learned about them when I got to Mount Sinai.
It's amazing we learned this. Yeah, so that's a cool story that an RNA virus can activate a DNA sensing pathway.
Yeah, with the dengue. But our stuff is VP35, do not block it.
No,
so that's like another way if we want to target the virus as well. And then I think on the same lines, we also have other assays in the lab that we're developing at the time to target the virus, and that was the mini genome assay. And it was also a luciferase-based assay that was optimized in a 384 well format. and we ran. I think that's the one assay that has gone way beyond. I don't know, Chris, we have screened over 400,000 compounds by now.
I mean, at least that many
Yeah.
at this point, and that assay at least
Yeah.
identifies compounds with real antiviral activity. So it's a bit more promising.
So is that something you're interested in continuing? The antiviral screen for Ebola viruses? Is that?
It's definitely going on. I think a lot of effort right now is to follow up on some of the scaffolds that we have identified and then do the mechanism of action studies. But it also depends on how the grant situation is, and Chris can tell more on that.
But it's something that we're still pursuing, and I think we'd like. I mean, this idea of a small molecule is targeting the replication complex, which should be relatively conserved across the family, is appealing because maybe you get a pan-filovirus inhibitor that would do not just Ebola but also the Marburg side of the family. So that's.
I guess an inhibitor would be still clinically useful, right? Because currently all we can do is do ring vaccinations, right?
And I mean, there are clinical trials going on in Democratic Republic of Congo now. There was a trial for different treatments. 3 of them were antibody-based. One was a small molecule nucleoside.
Right.
And the two of the antibodies were showing a clear benefit above the other treatments. And so those are the ones that are being pushed forward now for treatment. I don't think that data, I'm not sure about whether the data says the small molecule is or is not effective, but I think there's certainly room for additional small molecule candidates.
What was the name of that? Is it?
Which one?
The one that's being tested in DRC.
The antibody?
The small molecule inhibitor.
It was the one of the Gilead stuff was supposed to go there. That's the GS.
Remdesivir.
Oh, it's GSK something. Okay.
No, it's the GS. It's the Gilead.
Gilead. Okay.
I think it's GS.
I don't remember the number.
Yeah.
It has an official name, but it's tip of my tongue.
I mean, I can look it up on my phone if we need to.
This is
remdesivir, wasn't it?
Yes.
Remdesivir?
Yeah, but it does everything.
I mean, that's a ribonucleoside analog. And so it's very broad spectrum. Of course, needs to be phosphorylated, converted to the triphosphate intracellularly, and then can block viral replication. And so this is broad spectrum. I think it was included. Of course, Ebola. There is some desperate need for effective antivirals, but in this study, as Chris said, I think that the biologics, so the antibodies, came out more efficacious.
Yes, it's for sure. Yeah. So there's clearly room for more small molecule.
Yeah, no, absolutely.
Right. Yeah, well, funding is an issue with this. I mean, who's going to pay for it, right? So far, NIH or NIAID has done some, right? But it's not clear if that's going to go on forever, right?
Well, and it's often that famous valley of death of drug development, right? That NIH is very good at the early stage, and so early screens, assay development, and then funding, usually private industry funding, is there for very late stage. But there is this broad spectrum of formal preclinical development and early clinical trials, which require increasingly large amounts of money, and really there is no established mechanism.
Right, that's true, and that's the only reason the Ebola vaccines were tested, because we had the big outbreak in West Africa.
Right. Yeah. No, I mean, the platforms had been available for about a decade, and it took a crisis to push them into people.
Yeah, it shouldn't. It really shouldn't. I mean, there's a group put together by Kartik to develop monoclonals against other. Not Ebola, but other Hantas in Crimean Congo, where there may be a need. Yeah. So we should be ready, at least bring something up until a phase 1 and then have it in case. And that seems smart, right?
No, I think that's the way to go. And I think, that's the way that I think the NIH has thought about it a long time, but things tend to stall.
Well, I think part of it is there are political considerations, right? If there's no threat to the U.S., it's harder to get the money for it.
Sure.
Only when Zika came into the U.S. did we get money, which we took away from Ebola, if I remember correctly.
Yeah, that time. Because 2014, that was the time when everyone was speaking out about Ebola, and then we got
so there was money
cases.
For Ebola, and then it wasn't all spent, and some of it went to Zika.
Right.
So this seems a crazy way to to do science, to have any political considerations. It should just be about, being ready. So we're not going to solve that here, but clearly you and others are interested in being ready for certain virus infections, and it's clear that we'll always have Ebola infections. And in fact, that's where I met you last here in Atlanta, was at a microbiotics dinner.
Yes, that's right. Yes.
Because you're collaborating with them on
Yeah.
these screens, right?
That's right. Yes.
And we've also started collaborating on enterovirus antivirals for a relatively rare enterovirus infection, but nevertheless, the parents whose kids are paralyzed,
Yes, they care.
they
Right.
care. And so I have to tell you this, I wrote a grant proposal to enterovirus D68, 500 cases of paralysis in the U.S. since 2014. And one of the reviewers said, there's no medical need. I said, tell the parents that, right? But anyway, what if it gets bigger too?
Sure.
Are you continuing this, the immune suppressor work? I mean, where's it going? You haven't solved everything, right?
So one of the real interesting things is I said the Marburg virus VP40 protein blocks JAK1 function.
Right.
The other interesting point about VP40 and Marburg is that it seems to be a host range determinant.
Oh, right. Yes.
So to allow the virus to cause, this is true for Ebola or Marburg, to cause disease in a mouse or a guinea pig, you have to serially passage the virus.
Right.
And it accumulates mutations, and then it acquires the ability to kill the animal. In the case of Marburg virus, you consistently see mutations, amino acid changes that accumulate in VP40. And we've shown that you can take the parental wild type VP40, it blocks interference signaling very nicely in human cell lines. It is much less impressive in mouse cell lines. But if you take the mouse adapted version of the VP40s, now they've acquired the ability in multiple cases to block signaling in the mouse system. So we think it's important probably for virulence as well as having this interesting function, but we don't know how it works. So we're very interested in trying to understand what is VP40 engaging within the cell to block these pathways. Because we don't think it's a direct interaction with JAK1. At least we've never been able to demonstrate that.
So this is an example of how you can use this virus and its proteins to understand these pathways.
Yes, yes.
Not necessarily make therapeutics, but understand basic signaling in the interferon pathway.
Yeah, I think from a scientific perspective, if you can, reveal new basic cell biology, that would be maybe the most exciting thing you can do.
And so you can do most of this here because you work on proteins.
Yes.
And you put them in other viruses. And whenever you need to use the actual Ebola virus, you go to Galveston, right?
I mean, we go to Galveston, or we do a little work with Rob Davey now, who's at Needle in Boston University.
That's right, because that's now functional, right?
Yes, we still have the in vitro side, and I think the in vivo side is coming.
So they don't
soon.
have animal work yet, right?
Last I heard, they didn't yet have animal work up and going.
Okay. Yeah. And Priya, what are your future interests? What are you going to be doing?
So I think I'm going to still be catering the theme of virus-host interaction. I think one stuff that we didn't mention. So we did like a big Ebola viral protein screen, the mass spec screen, in collaboration with Krogan Lab. And that work was out like last year.
To see which cell protein?
Which host protein. So just to see like the host makeup of like in the virus life cycle. Like
so did you do all the viral proteins?
Yes, all the Ebola, except for we couldn't get L, because that didn't express well. So this was all the Ebola viral proteins. And the stuff that was published was with VP30. It was like a former postdoc in the lab who did that. So I've been following up a lot of host interactors of VP35 proteins. Right. So that's like one of the things. I mean, it is like telling us a lot of stuff that we already knew, but we never had evidence. And then a lot of things, so that is like another study that I've been working on, hopefully wrapped up soon. And we found like some cool interactors, and hopefully that can help us get around like how like the virus behaves in like the host cell, and what are the key pathways that could be targeted in future.
That's what you were talking about, seeing what these proteins are interacting with.
Right, yeah. And it's interesting, that data set, if you look at sort of common interactors that targeting specific pathways, you see things coming up that you wouldn't normally associate with a negative strand RNA virus that replicates in the cytoplasm. So again, we're excited that it will reveal some novel biology we didn't appreciate previously.
I guess you have to look beyond our biases, right?
No, absolutely, yes. Yes.
This is a cytoplasmic virus, but maybe there are nuclear functions involved.
Oh, for sure.
Right?
Yes.
So for picornus, for polio anyway, this may have been done at Einstein in the 50s or 60s. If you take the nucleus out of a cell, the virus will still replicate. So for years, people said you don't need the nucleus, but we do actually. Things that are coming out. So are you going to continue to work on these interactors? What does your future have?
I mean, so some of the interactors are like common to like other viruses as well. I mean, so there's like part of things. But I just recently got an offer from Trudeau Institute. So I will be like joining them soon in like spring next year. But the hope will be they want to start with like the whole program with the virus immunity thing. And then part of the same history of things with the virus was interaction, but also moving on to tick-borne viruses. That would be like one field of things, but looking into the similar angle therapeutics and like the mechanism of actions and the host interactors.
The Trudeau, that's in upstate New York, right?
Yes, up in Saranac Lake.
A little bit of a change here.
A lot of change. A lot of change, yes.
Are you still going to interact with Chris in his lab, do you think? Or?
Yes.
I think we'll continue to work together, yes.
Yes, it's like one of those, I think me and Chris have talked about so many times, like a lot of things that we find is like, you see there are so many Zika screens and Dengue screens that are coming along. So it tells you something, why you have so many common interactors with all these viruses, which have nothing to do with each other. So there has to be a common theme behind it. And if you can find really a way to target these things, you might have your golden bullet. You know, that would be the hope.
Well, I guess the ideal would be to have an inhibitor of all RNA viruses, and then you'd have no trouble getting that tested by companies, right?
That's correct.
I'm not sure we could ever get that, but I'm sure it's maybe worth looking at, right? Because even if you look at a polymerase, they're all slightly different, so you wouldn't be able to get a pan-polymerase inhibitor, right? But this nucleoside analog you mentioned, is it toxic for the cells at all?
I mean, that's a loaded question with every antiviral, right? So, of course, I mean, every drug has a toxic concentration. And sure. So the problem is selectivity for the viral polymerase. So does the viral polymerase recognize the analog, but host polymerases do not, right? Or host polymerases, whatever damage is done, can repair it, right? But because the RNA virus polymerases do not have any repair mechanism. But then what we see a lot in the field is that obviously mitochondria also do not have mRNA polymerases do not have a repair function unlike nuclear, and so mitochondria toxicity is usually the first problem. And so that means, of course, I mean, so it's always, the question is, what's the selectivity index, right? So what is the effective concentration versus the toxic concentration? And that is, unfortunately, with many of the nucleoside analogs, that window is small. So those concentrations are very close together. And then often side effects are just unacceptable. But of course, it also depends what is the disease you're targeting. Right, and then so my lab looks a lot at influenza, and in particular seasonal influenza, and then of course the drug safety profile that we define what is like a hit or a clinical candidate that we think has potential to be moved forward. So obviously much more stringent what we tolerate in terms of off-target effects or side effects than for example for an Ebola therapeutic.
Right. So is there a need for additional influenza antivirals?
So I think with influenza, when we started that, really, what we've seen in the past year is for really a couple of decades that the first new class of antivirals against influenza was licensed, baloxavir, maboxil, or Xofluza. Which is a new class. So we had before the neuraminidase inhibitors, and so this now is an endonuclease inhibitor, so targets the polymerase complex and prevents cap snatching. So essentially prevents the formation of viral mRNAs. But the problem for both, so what we know for a long time for the neuraminidase inhibitors is viral resistance. And that of course in the world of small molecule antivirals is always the the ongoing topic. An antiviral that does not cause resistance is not an antiviral. Was the old word, and I think it still is true, of course. The question is there a penalty for the virus in terms of viral fitness or pathogenesis penalty? So how important is resistance in the field? But what we know with the neuraminidase inhibitors is that increasingly, for example, with the 2009 pandemic, swine origin influenza, that several of the circulating strains actually carried already resistance mutations. So essentially you have pre-existing resistance. And now after really one season with Xofluza, what we see is that resistance again emerges very quickly. And so that is really the problem. And to answer your question, I think really the next generation influenza virus inhibitor needs to come with a high genetic barrier against resistance, against viral escape. And so that was part of for our compound that we have that we favor. That's also a ribonucleoside analog. That was part of the main developmental exclusion criteria, that we do not see rapid escape. And so we just have a paper coming out shortly in Science Translational Medicine that describes efficacy of this compound in a ferret model, and also in ex vivo human airway organoid cultures. And in particular addresses the resistance problem, or the resistance question. And what we've seen is, in biology, you can never say never, but clearly the barrier is very high. So we have so far not seen any robust resistance, but we have seen oral efficacy and significant duration of disease symptoms, in particular in the ferret model. So therefore, we think that obviously we are very excited about our compound that will enter the clinical phase next year. But we feel that really the next generation of influenza drugs need to have this as an inbuilt property, that we have a less rapid viral escape.
So, did you design the screen in some way to exclude or to enhance compounds that would, if you got resistance to them, there would be lower fitness in the virus?
Yeah, that would be nice if you could proactively design the screen. So what we did is, in terms of screening, so I do a lot of screening in my lab. I have set up automated facilities, and we also screen open discovery libraries. So we talked about small molecules. That is what we screen. And then for this project, ribonucleoside analogs. But I think it depends on defining the right drug profile. So what is actually the therapeutic that we want to have at the end? What features does it have to have? And that needs to drive the screen. So I feel, at least when I entered this, I thought it exactly the other way around. And I feel, talking to colleagues, that we a lot in academia, we do that a lot, that we think it from the front end. So we have the virus, and then we have a small molecule and we see what it does. And then we try and move it forward and find at the end a disease indication for it where we can use it. And I think if you really want to increase our chances to bring something to market, so to develop an applicable therapeutic, we really need to think it from the end stage. So from the patient population, the needs of the patient population. And that we then move all the way forward. Okay, if we want to arrive here with our drug profile, then these are the counter screens we need to implement. And then we also need to be tough, even if we like a hit candidate, and very quickly people take a liking to their compound. I mean, we see it all the time in the lab. And then it's increasingly difficult because people get invested. It's increasingly difficult to let go. But I think one really needs to be tough there. And so that is what we did early on. So we designed counter screens that early on test for, okay, how rapidly does the virus escape? And then one really needs to abandon some that we think may look interesting, but not for our ultimate profile.
What you mentioned is actually a problem with science in general. People get invested in their theory, their hypothesis, and if they show it's wrong, they don't want to admit it. They keep doing experiments to work around that. This is well known in not just antivirals, right?
couldn't agree more. But the antivirals field, I think, has really as a particular history there, because a lot of the of chemotypes, so of small molecule structures or substructures that we very well know are just not developable. They are not drugs because they aggregate or they are chemically reactive. They covalently engage protein targets. So they have features that are just non-desirable. A lot of that has actually been worked out in the pharmaceutical industry for quite some time. It is actually in the public domain. And so what academia then has done when cost for high throughput screening and for libraries came down and academia really discovered drug development, that we now, with public money, have actually collectively in the field repeated many of those steps and rediscovered with great effort that certain chemotypes are just not developable. And I think that is, so it's very difficult to convince people to let go when it looks good initially. But I think in the interest of having something that is developable, that can advance, I think we need to be tougher with our pet compounds.
Yeah. So you reference something that I noticed in this paper called "Biology Must Develop Herd Immunity Against Bad Actor Molecules."
Yes.
So the idea, drug discovery was originally the domain of the pharmacy, but academic researchers have increasingly entered the field. So what do you mean by a bad actor? Is this something you just mentioned, that you rediscover the same compounds?
Yes, so bad actors are so we call it in the field assay paints, that are pan-assay interfering substances. And so that are compounds, when we run high throughput screens, that are compounds or chemotypes that come again and again.
I see.
So against multiple assays, so they are pan-assay interfering and enormous, and so now we can break this down into something like the real bad actors is maybe 11 or about 11 chemical classes, and then of course you get that they come in different flavors, right? So they come with different substituents that they may look at first different, but if you dress it down to what's the core active substructure, then you have one of these.
Okay.
And they have been really a pain in the field. They have cost us collectively enormous resources, financially, but also people resources, time that has been spent, and they are just not developable. And some have even advanced to clinical trials, to clinical testing, but then they usually failed. And that is these famous collections of confirmed bioactives,
right.
Those libraries, many of them, not all, but many of them are really enriched for these compounds that have made it, that looked good initially, and that have then stalled at some point in the development, but made it into those confirmed bioactives.
Yeah. Now, why has drug discovery shifted? Because we've heard today for different viruses doing screens, right? You guys have done it for Ebolaviruses. Why are academics doing this more and more? Whereas the drug companies used to do this. Do you know why?
Yeah, so that's an interesting question. I think for academia, one aspect is really, as I said, that cost I think really came down a lot. So in the early 90s, to set up a true what was high throughput at the time, and we're not even talking ultra high throughput.
Yeah.
That I think would in most academic settings, certainly for individual labs, would have been completely out of scope of what was possible.
I would offer the explanation that basic scientists are increasingly being pushed to do translational work. And I think sometimes this can be misguided in that some of the most fundamental and important advances have come out of very basic studies that weren't really trying to solve a specific problem or develop a drug, but just do very basic investigations into biological systems. But increasingly, it's getting harder to get that type of work funded. So I think there's an incentive for academics to try to do something more applied.
Yeah, I think that's probably important as well. Yeah, and that begs the broader question of who should be doing this. I mean, shouldn't university research be basic science, right? That's what it traditionally was, and seems to be changing.
But I mean, how much? I think the way I see certainly modern drug discovery and drug development, it has to be an interdisciplinary effort.
Yeah,
and
yeah.
so I do think really, because from pharmacology to very basic virology to clinical knowledge and clinical trial design, I think one really needs to span such a broad range of areas of expertise.
Well,
I think collaborative, and also collaborative between academia and industry, what we see increasingly.
I think that's a good point. That's why your collaboration makes a lot of sense, right? Because the company will do the screens, and then you can benefit, and they can benefit. It makes a lot of sense.
And then you also, the companies will have people with the type of expertise that Richard is referring to that we don't come with.
And then, of course, if they do a phase 1, they will not, depending on the size of the company, they probably can't do it, but they can work with other companies and do things that academics not normally would be involved in. Right?
I mean, that's the hope, certainly.
Yeah.
We get to that point, yes. Richard has actually managed to get to that point.
So I wanted to ask you, Richard, for HIV, the key was to use 3 drugs.
Yes.
Why don't we do that for influenza? We have now 2 classes, right? Has anyone tried a neuraminidase inhibitor plus the endonuclease?
Yeah, combination. So
it also worked for Hep C, right, as well? Yeah.
Of course. And that may be really the answer, or another answer long term. But as you know, if you want to obtain original approval, original licensing for the drug, then it needs to be tested on its own as a monotherapeutic.
Right.
And then the combination requires another test.
Right.
And those tests, again, these are clinical tests, so we are talking about enormous investments. Mm-hmm. So therefore, if we start out with a drug that first would require testing as a monotherapeutic, but we actually have very little hope that it would be suitable for monotherapy, then it will be difficult to obtain that funding for the very future prospect that maybe in a combination it will be actually very beneficial. So I think that is really a developmental hurdle that would need to be overcome. Or we need to look initially at drugs that have efficacy for monotherapy and can then be combined if required.
Right.
And I think it's really, compared with HIV, is the question, if you look in particular at these more acute RNA virus diseases like influenza.
Right.
How much will a triple therapy be required? I think probably not. So we may really HIV and the persistent infection, I think, causes
That's
a
a good
challenge.
point. So if you have enough of an arsenal for influenza, if there is resistance, you can just use something else, right?
Yeah.
Instead of combination. That brings up the interesting question, since it is an acute infection, you have to treat pretty quickly after.
Yes.
And we don't have any good, as far as I can tell, there are no good rapid diagnoses for flu yet. There's one antigen-based, but it's notoriously inaccurate. So doesn't that have to advance as well?
Yes, and the awareness, right? Because at least talking for myself, I need to feel pretty bad before I would go through all see a healthcare provider, get myself tested, and then obviously often that window of opportunity for treatment, for efficacious treatment, may have closed. Absolutely. So that's a very very good point, and I think so for influenza is probably really in the first 24 hours after the onset of symptoms. And I think then with every ongoing hour, the benefit for the patient of treatment goes down. But for some of the other major targets, in particular the pediatric targets like respiratory syncytial virus, RSV, that's also major focus, major health threat, and major focus for drug development now for several years. But so far we really don't know in the field how wide this therapeutic window, so the time window for efficacious treatment really will be. So I think that is, we need to have the efficacious drugs to actually really explore what is the impact in the field under real world conditions, and not experimentally controlled or clinical trial with controlled conditions.
So basically, once you have effective antivirals, then
Yes.
you'll answer the question of how long can we give that, and then better diagnostics will come only once you have the antivirals. Probably.
I think it will clearly drive this, because if we know that we can actually make it different with
sure.
treatment, then that is a major incentive to, and also to implement available rapid diagnostic tools to implement that more frequently.
I'm sure it can be done, right?
Right.
Whenever there's a need, technology.
Right. But at the moment, I mean, from a healthcare provider perspective, if it's clear, if the disease is viral, we really have only supportive therapy, but we do not have an effective antiviral, then the incentive for molecular diagnostic is much lower than knowing that we actually would have something at hand that will benefit the patient.
So you mentioned respiratory syncytial virus, very important in very young babies.
Yes.
And the only therapeutic we have at the moment are monoclonals, right?
That's right. So palivizumab is a neutralizing monoclonal antibody directed against the RSV, so respiratory syncytial virus fusion protein. Mm-hmm. And palivizumab is efficacious at the moment, really preserved for high-risk patients. That is mostly infants born prematurely or with underlying additional complications, for example, congenital heart disease. And there, where RSV infection is life-threatening. And so palivizumab is efficacious, but the problem really that prevents its broader use is cost. So estimates are that it's about to prevent one hospitalization due to RSV with palivizumab is roughly $300,000, and that is major. So, and here we are talking about patients in the United States, but the disease problem is more global. And obviously many low and middle income countries, a therapeutic like palivizumab is just not
Sure.
on the cards at all. And so in the cards. And so, therefore, we think really that biologics have the advantage, like antibodies have the advantage that we understand them quite well. Safety profile is usually very good, but the issue is really cost. While small molecules have really the strength that many of them can be produced very cost effectively. They are shelf stable. They do not require cold chains, and so it really. Ideally, if money wouldn't be a problem, we would have a bit of everything. But I think if we look more for the global need, then we also have to address the cost issue.
I remember, I don't know if it's still done, but you used to use ribavirin for RS. It's not used anymore.
So officially it's the only small molecule that is licensed, officially approved for use against RSV, but ribavirin has terrible side effects.
Yes.
And so it's really not used, certainly not in infants.
So you have published an inhibitor of RS, which blocks the RNA polymerase, correct?
That's correct. Yeah.
And is that going to go to phase 1? Is that the plan?
So this is for RSV. For what goes into phase 1 now is an influenza.
Influenza is the one we talked about before, right?
Right. That's correct. This RSV compound is at an earlier stage in development. That is not ready for formal development yet. And it's really with an antiviral. So what drives efficacy in vivo, first in an animal model, right? And this is most of what we are doing in the academic antivirals world. And I think is really, of course, the direct potency. So what concentration do we need to reduce virus replication by 50% or by 90%? But what is equally important is also the pharmacokinetic properties of the compound, and that often gets, at least in academia, forgotten. So if the half-life in a living organism is 2 minutes, then the lowest efficacious concentration will not help us much. And so this compound for us at the moment sits at the stage that we like potency a lot. It has decent PK properties, but we want to see ideally both or one of those two parameters to be a bit better before we think it is ready for formal development.
So that involves modifying the compound?
That's
right?
correct. So my lab has collaborated now for over 10 years with synthetic chemists at Emory University. So that started when I was with my lab at Emory, and we've continued that. And so that's really, these are medicinal chemists who can synthetically change, alter initial hit candidates. And so we call that, we develop structure-activity relationships, or SARs. So we modify the chemotype slightly and then test its effect in the bioassays. And so then we start to understand how chemical properties and activities are related. And once we have that, then we can identify features of the compound, of the chemotype that need improvement. Let's say it's highly hydrophobic. That usually, if you want an orally available drug, that usually is going to be a problem. But if we know where a chemotype tolerates changes and where it interacts tightly with, let's say, its protein target, and therefore cannot be changed at all, then we can design then our clinical candidate by making informed changes.
And if you wanted to inject it, say, in a population which couldn't take it orally, very sick people, then you would have different. A hydrophobic drug might be acceptable.
Maybe more acceptable, but also there. So for IV formulations, that's usually the most challenging because it really needs to be fully dissolved. So orally, you can often give as a suspension. And then if you think that if solubility is often pH driven, and so with a very low pH
the
stomach, that works. But if you have a suspension and you give this IV, then not feasible.
Yeah, that's where collaborations, as you mentioned, are important because you're not a chemist, right?
I'm originally a biochemist, but that is true. So I am not a synthetic chemist.
Yeah. So collaborations are important with chemists and in order to do this.
Right? What I referred to earlier, that really drug development has to be an interdisciplinary effort.
So let me ask you one more thing. You've also worked a lot on the measles RNA synthesis.
Yes.
Are you interested in developing antivirals for measles? And if so, do we need them?
So for measles, it seems to be more antivirals seem to find me than I'm looking for them, and I don't know why that is. But so we have developed. This is the drug that we call in the lab the drug that we originally never intended to develop, but it just never gave us a reason to kill it. So typically when you have a new hit after a screen, then nobody wants to waste time. And so we look for the first reason that the compound actually fails a critical test, and then we can kill it and move on. And so this compound that came out was active against measles. And it really, as I said, we developed it because, or we moved it forward because it didn't want to die. And so we, in a collaboration with people in Germany, the von Messling lab at the time, we tested it against the canine distemper virus. That's the canine closest cousin of measles. And also more recently, that's a paper that we're just writing up in a primate measles model against the clinical measles isolate. And this compound is orally efficacious, and it completely prevents disease when given during that incubation period post-infection, but before full onset of measles symptoms.
Like rash?
Like rash,
okay.
Yes. But and so then it completely prevents all disease and all clinical signs of disease. And the treated people, and in case of CDV, we use ferrets, and CDV is fully lethal in ferrets. So then the surviving ferrets that come out essentially as vaccinees. So they develop a very strong adaptive, for morbillis in particular, humoral response. So that is all very encouraging. But the question is really, do we need a measles antiviral? And I have been discussing this now really we collectively in the field, also with people at CDC and WHO, because as you know, we have the measles initiative, and measles is, at least in principle, considered to be a virus that can be eradicated.
Right.
And the problem is really, as we have experienced just in the past year in the United States, and as we experience in Europe continuously, that against measles one needs an extremely high herd immunity. So essentially, everyone needs to have 2 doses of the MMR vaccine, or we do not get this herd immunity required to prevent outbreaks. And we are just here localized in small geographical areas in certain communities. We are below this herd immunity.
Right.
And in Europe is more of a broader problem that overall the herd immunity is maybe depending on the area between 70 or 80%, but not above 90, but it has to be. So do we need antivirals? The question is, so talking to some of the measles people in the field who are actually responsible or involved in outbreak controls, for example, the team from the CDC, would they have liked this year in the New York outbreak, would they have liked an antiviral? Yeah, very much.
Sure.
But can we mobilize the resources required to develop a drug that, from an industry point of view, is commercially really a very, very unlikely opportunity. So it needs to be public funding.
Exactly.
That is a major challenge. And the second challenge, so we thought it through, at least to some degree, is really how would we design a phase 2 clinical trial that with measles would need to happen in pediatric patients. There are major ethical problems associated with that when really there is no blueprint. One would need to establish this as one goes. That's a major challenge.
Yeah, because we have a vaccine and you can't deny that, right?
Exactly.
You have to,
exactly.
With any treatment, you have to give the standard of care, right? So you can't deny what's available. But I mean, the question is, if you have a rash, is it effective? The antiviral effective? You said no. Right.
So the paradigm is that measles is essentially immunopathogenic, and so that means that the rash, the clinical science Diane Griffin has done decades ago. She first published excellent work and on the correlation between clinical signs, so rash, fever, and viral titer. And what that demonstrates is that viral titer already drops dramatically at the time of rash. Right. So what could an antiviral do under those conditions? But what we would like to address, and we will address with using, again, CDV and ferrets, is whether we can suppress or at least alleviate long-term problems of these viral infections, in particular measles. A lot of the clinical complications arise from strong immune and lasting immune suppression of the virus. Right. That is for several months after infection, resulting in secondary bacterial infection that, again, in particular in low-income countries, is a major clinical problem and resulting in case fatalities, more than primary measles. And so the question is, if we with an antiviral that prevents also the very low ongoing virus replication or the persistence of viral RNA, can that actually shorten the duration of immune suppression? And if that is, so that I think is very testable in the CDV ferret model. And if that is possible, then I think that is a major incentive to actually develop a measles antiviral, because then there is major benefit for the patient.
That's a great point. Yeah. So you may end up having an antiviral you never wanted.
That's right.
That's really interesting. Well, I frankly think that we could use a new vaccine that is free of all the Wakefield contamination, because I think that lasts forever and we can't get rid of it. But that would be even more challenging. But can you imagine a subunit vaccine, not infectious, put on with a microneedle patch?
Yes. The patch, I mean, subunits, as you know, all the RSV and measles, the problem with the vaccine development is you do not want to cause atypical disease. So the inactivated vaccines that then resulted when...
Yeah, sure.
Right. So that is always the concern. But microneedles is, I think, is very interesting and could also overcome the fear people have of the needle.
I think a lot of people do, and they use that make other excuses because of the needle fear. And I think overall, if we could convert all injected vaccines to microneedle patch at some point, it would really go a long way. But that's some time off, as you know. Okay. All right, let's talk with Andrew a bit. You published just last week this paper, Segmented Filamentous Bacteria Prevent and Cure Rotavirus Infection. This is in Cell. And I'd love to hear how a non-virologist got to this story. What led up to it?
Well, years ago, I was focused on bacterial interactions with intestinal epithelial cells and doing a lot in cell culture systems and mice, just trying to understand those basic interactions, how the immune system, particularly the innate immune system, can recognize bacteria, distinguish them from all the commensal bacteria in the gut microbiota, and the role this played in infectious disease and chronic inflammatory diseases. And then I saw Mary Estes give a talk at one of the GI meetings on rotavirus. And I thought, wow, this is really interesting. This is a very important virus. It's killing hundreds of thousands of people. And it might be a great opportunity to see to what extent did the findings we had made regarding how the immune system recognized bacteria, how this would apply to a virus like rotavirus. And we were getting at a lot of those questions, and one simple way to, since rotavirus is normally efficiently cleared by the adaptive immune system in mice and humans. So in order to focus on innate immune responses, we were using immune deficient mice, RAG mice that have no T cells or B cells, and they're used as a model of chronic rotavirus infection. So this is the model that we were using, and then unintentionally, we developed a colony of RAG mice that could not be infected with rotavirus. So initially, we had bought these RAG mice from a major mouse supplier, Jackson Labs, and they would develop chronic rotavirus infections when we inoculated them, as had been described by Mary and others. And then once we were breeding those same mice here at Georgia State, they became uninfectable. So wasn't something we went out looking to do, but we just thought this is really interesting and maybe...
So completely uninfectable.
Completely uninfectable.
Not even an acute infection.
Not even an acute infection. So mice that, a strain of mice that would normally develop a chronic infection and that they could never clear, all of a sudden we just couldn't infect them at all. And that's really how we got into that area.
So you decided to figure out what was going on in those mice.
We did, and our thinking was, so we had a guess initially early on that it might be differences in the microbiota, that perhaps they had acquired some organisms here that were making them uninfectable. And we thought that maybe if we could understand how this was working, it might give us some basic insights into why there's such a varied susceptibility to rotavirus around the world. So that was sort of the logic, and the basic goal was if we could understand why these mice were uninfectable, that maybe we could learn something.
Okay. And if you got fresh mice from Jackson, they were infectable.
Yeah, and what we figured out fairly quickly after that is if the student leading the project, Zenda Shi, if he did all the breeding and all the cage changes and everything himself, he could maintain them as infectable and see the previously described phenotypes indefinitely. It was only when the animal staff here were doing the husbandry that the mice very quickly became uninfectable.
Okay. It's a common story. Something is going on with the animal handlers, right?
It is. And then subsequently, as we were investigating this, we did realize that our colleagues who became our collaborators at Washington University in St. Louis, that they had observed something similar, that they had a colony of rag mice that they had even removed on top of the rag genes. They had removed the genes for type 3 interferon, which had been thought to play a role in controlling rotavirus. And even those mice remained uninfectable. So we realized that there might be a pretty common tendency for mice to acquire microbiota that make them uninfectable by rotavirus.
So then you went through a series of experiments. You showed that it was, if you co-housed mice, you could transmit this, right?
Yep.
It was through the feces.
So we could transfer the uninfectable phenotype by either co-housing or by a fecal transplant. And importantly, we could do this with a fecal transplant where we wouldn't just administer fresh feces, but we would make suspensions of the feces, glycerol stalks, put them in the freezer, and realize a thawed aliquot could still transfer this uninfectable phenotype. And from my perspective in the lab and advising a student, that's when I realized we can go to the next step. We can figure out exactly what is conferring this uninfectable phenotype. Whereas if it's something that you can only transfer by co-housing, it can be pretty tricky to ever solve that, since there's so many different types of bacteria in the microbiota.
You can't just sequence the gut microbiomes of both.
Well, you can, but you'll get thousands of differences, and it's very, very difficult to figure out which ones are responsible for the difference.
So you developed a really unusual way to identify this, right?
Yeah, what we ended up doing, so we took those frozen aliquots and we did everything we could to destroy everything except what was transferring the uninfectable phenotype. So we realized that we could heat them to kill off the non-spores, treat with a variety of antimicrobials. But even that, after we transferred it to mice and made them uninfectable, there were still too many bacteria to figure out what was going on, since the mice we would transfer them to already had a lot of bacteria, and anytime you do a transfer of this sort, you change those relative populations. So what we ended up doing was taking germ-free rag mice that had no gut bacteria at all, and then doing a limiting dilution transfer. So diluting our sample down and then looking for which would allow us to obtain some mice that had acquired the uninfectable phenotype and some that had not. And then doing the sequencing on those, then we got a very clear and striking result that the mice that had been protected, that correlated perfectly with transfer of one specific bacterial species, segmented filamentous bacteria.
So when we have a faculty in our department, Evalio.
Yeah, I'm very familiar with his work.
Who came from Dan Littman.
Yep.
Who originally, I think, made this discovery. He was buying mice from different vendors, and some had Th17 cells and others didn't.
Yeah.
And it turned out that it was SFB that was the responsible.
And we were aware of that work. And when we got SFB, we thought, well, initially this seems strange, since this was rag mice and they don't have any lymphocytes, let alone Th17s.
Yeah.
And then we went on and even deleted IL-17 or neutralized it with antibodies, and that had no effect. So this doesn't seem to be an impact of Th17s, but I think what it's showing is that SFB closely associates with the epithelium and is having a variety of impact on host phenotypes, and Th17s is one of those impacts, but it's having pretty profound impacts on host gene expression. And it also seems to have direct effects on rotavirus and some other viruses as well.
So SFBs are not easy to culture, right? But you figured out you could culture them in mice. Right?
Yes. So it's really hard to culture them out of a live organism. A few attempts have been made, but they're using media where they're taking components from animals, so it's very hard to get anything reproducible. But what you said is exactly right. We're using the germ-free mouse as the ultimate culture media. Anything, nature will find a way that if you put one organism into a germ-free mouse that has the potential, any potential at all to live and colonize, I would say that there is no broader spectrum, more open culture media than a germ-free organism.
So you can
like
give
a mouse.
germ-free mice SFB and only SFB,
Right.
and then the fecal material has just SFB in it, right?
That is correct. Well, the SFB be the only live thing that's in there.
Okay.
There does tend to be a lot of dead bacterial DNA in feces, since there's bacterial DNA everywhere, and even autoclaving it won't destroy it. But SFB is the only live thing that's in there.
And then you can feed that to rag knockout, and it will protect those mice.
That's exactly correct.
So what do you think is, I know you're not a virologist, but I'm sure you're thinking about how it's protecting against infection.
Yeah, so we're definitely working on this, and at the moment we have identified 2 potential mechanisms, and we were grateful to Cell for letting us get by with the idea that we have 2 potential mechanisms and not really making us choose between them. and the 2 mechanisms are one, that it can have direct impacts on the virus. And in fact, Richard has helped us out with that in his screens, testing some other viruses, and it seems to have those types of impacts, at least in vitro, seem to extend to other viruses.
Outside of the family of rotavirus.
Right, outside of rota. So as an intestinal virus, it also impacts reovirus, and then Richard and his screens found some impacts on influenza and VSV as well. And these aren't necessarily going to be physiologic interactions. This is really just showing the point that the other thing about the microbiota is it can be quite a source of a variety of different chemicals. So that within this vast chemical ecosystem, there may be a variety of ways to impact other viruses.
And you also think it's the bacteria doing something to the epithelium, right?
So one thing that the bacteria does, and this actually even goes back to Ivo and Dan Littman's work, is that by directly interacting with the epithelium, it increases epithelial proliferation. And this will give an increase in epithelial turnover. So in the case of rotavirus, it is uniquely infecting villus epithelial cells, so the cells at the tips of villi in the intestine. And these cells are relatively short-lived cells. The average intestinal epithelial cell lives a couple of days. They migrate from the crypt of the intestine up to the villus, and then they're shed. So if you imagine an escalator where cells at the top or something at the top of that escalator is being infected, and though the virus is maintained, particularly in a rag mouse, by going from one cell to the other quicker than those cells are shed, that if you simply speed up that escalator and make cells slough off faster, that we're thinking this could be a very ancient mechanism of immunity. And in the case of a rag mouse, or even we see the same impact of SFB in a mouse that not only lacks T cells and B cells, but also lacks innate lymphoid cells, that one of the last things, and rotavirus, like the other viruses you were talking about, is also pretty good at evading interferon. So one of the last mechanisms left might be just speeding up this escalator and making these cells turn over faster. So we know that we've been able to show that making these cells turn over faster can reduce levels of rotavirus. So we think that we're leaning towards this maybe being the more important mechanism in vivo, but it's a little bit hard to tell. So at the moment, we're pursuing both mechanisms.
So many years ago, we made transgenic mice susceptible to poliovirus. Just take the human receptor gene, put it in mice. Those mice, you can inject virus, they get paralyzed. But if you feed them, no paralysis unless you cross them to IFNAR, type 1 interferon receptor knockout mice. So maybe there are SFB, and you should check polio.
Okay. Yeah, no, that would make sense.
Makes sense, right?
Reovirus is a little bit like that in that it's an infection model in immune competent mice, interferon competent mice. And SFB does nicely protect against reovirus. In the IFNAR knockout, and this is collaborative studies we've did with Terry Dermody, who was really helpful in this study, that those mice, they'll eventually get a type of paralysis and die once the reovirus gets to the brain. So we know that SFB can protect in that model. So polio would be a logical thing to test.
I mean,
as well.
these are all human viruses. I guess, have you looked at a mouse virus to see if this protects?
So where most of our studies with rotavirus are done with the mouse rotavirus.
Okay.
Unlike these other viruses, though, the mouse model of rotavirus with a normal mouse virus and a normal mouse, the disease pathophysiology is very, very similar to humans. Young mice, like young humans, get severe diarrhea. Older mice, like older humans, get infected and shed virus. And they seem to have some GI distress. It's relatively mild, and we can't really ask the mice what they're feeling. But we think they have probably mild GI distress like humans would do, even without severe diarrhea. So in that sense, we think that it's a very good model. And this was one of the things that attracted us to study rotavirus in the first place, that it's one of the better examples of a mouse model that so nicely recapitulates the human disease.
Are SFB in wild mice?
SFB are absolutely in wild mice, and they are in pet store mice. There was a paper a couple of years ago that's gotten a lot of attention in the research world, which indicated that the immune systems of mice purchased in a pet store or captured in the wild is much more like a human immune system, whereas mice from suppliers like Jackson Labs, their immune system seems to be much more similar to newborn humans, indicating that these standard mouse facilities are not physiological. So using SFB as an example, it definitely falls into that category, that it's present in all of the non-vendor mice that we've looked at. So we think that our mice with the SFB are physiological, and that the mice one might buy from Jackson, we view those as the non-physiological mice.
What about people? Do we have SFB?
So there's a lot of work been done in this area, and a lot of it is from several studies from China, and they have reported SFB present in young infants, newborns. It hasn't been looked at in a lot of cohorts, and there has been some technical questions about the specific primer sets used and how reliable is the methodology. But we're taking a look at this. The first author on our paper recently started a position at the CDC, and he's now doing microbiota sequencing from fecal samples of rotavirus patients around the world, both to look, are there regional differences in microbiota, or are there differences within regions that correlate with those who had severe disease to mild disease. So our thinking is that we're not sure if SFB itself is going to turn out to be important in this scenario in humans, but that the basic paradigm may be applicable, that differences in basal microbiota may be a determinant into why in some places rotavirus seems to cause a very severe dehydrating hospitalizing disease. In other places, even though infection is pretty rampant, disease is mild. And the other important regional difference with rotavirus is that the vaccines seem to have been extremely effective in Western countries in reducing incidence and severity, and they've been quite ineffective in regions where they're needed most. And since the vaccine is a live, attenuated virus that has to infect in order to protect against subsequent infections, we think that microbiota might be a determinant of vaccine efficacy as well. So these are all a lot of the follow-up things we're working on.
That's a really interesting. It's often never as simple, but it's clearly important to look at SFB and rotavirus infections in people, and the vaccine efficacy makes perfect sense. So that'll be fascinating to sort out. I like this because I think this is the first example of a protective gut microbiota for a virus, because there are others where the microbiota facilitates infection,
and
right?
That's exactly correct. And even in the case of rotavirus, it follows the same path as these others. So rotavirus, poliovirus, a couple of others that have been studied.
MMTV.
They will infect less in germ-free mice. So a germ-free mouse is partially resistant to rotavirus. It will still be infected, but so I think what this really comes down to is to say that a microbiota promotes or inhibits infection is just a tremendous oversimplification, that it's really going to depend on the specific composition, and that some bacteria will aid virus infection and some will likely impede it, and the mechanisms are going to be pretty complicated. Some will likely be direct. In the case of polio, that the promotion of infection seems to be mediated by bacterial LPS binding the virus and helping with entry. And I think some of the effects will be mediated by the immune system. So I think we're in the early days of understanding these types of interactions. And I view our paper as an example that they can be very potent and effects that could be dissected mechanistically.
I presume you've had press interest on this paper, right?
Some. A lot of our other work has related to obesity. And I've kind of the last couple of days realized that the press is far more interested in people's waistlines than in children dying in Africa and India. So there's been some press interest, but not nearly as much as our work on obesity.
Because I'm sure they want to say, is this a cure for gastroenteritis, right? Because that's how they view it, as a simplistic outcome, right?
Yeah, I think that's about right.
But it's not always, as you say, it's never that simple. I got a call to comment in a story from another lab where they knocked out a gene in cells and it led to enterovirus not being able to replicate. They did it in mice. So they said to me, is this a cure for the common cold? And even though I said no, they still printed the story. And then someone emailed me and said, we can sell you a plaque of your story. It's not even my story.
Well, it has been trending on Twitter.
This has been the story? Yeah.
I know the NIH called us, the director, he tweeted.
He did, yeah.
Yeah.
Cool. That was recently. So yeah, I think he's very interested in
in great! You're going to get grants the rest of your life now.
We'll see. A lot of this work, my grants don't really directly relate to this topic. I could interpret grants on dealing with host microbial interactions with wide latitude to use the funding for this work. But
another example of how science can go in different ways that you don't expect, right?
Absolutely.
Very cool. All right. I have just two quick questions for each of you before we wrap it up. And Priya, if you hadn't been a scientist, what would you have done?
Oh, I think If I think about it, so I'm the first one to ever do PhD in my family. There's no one. I think my parents never thought I will be a scientist. They wanted me to be a medical doctor.
Okay.
So I think I would have gone into that direction if I have not been in sciences at all. Probably a chef.
A chef. It's like science, right?
It is. Like, I like cooking, the science of cooking. So
Do you know that Alt Lopez, the science of cooking?
Yes.
So he's the son of Fred Alt, who was an immunologist. You must know Fred Alt, right,
Yeah.
Andrew? He's an immunologist who was in the Baltimore lab with me, and his son is a science chef. Yeah.
Oh, okay.
That's very cool.
Good to know.
Okay. Chris, what would you have done?
I guess at one point I also thought about medicine, but decided that the basic science was more to my liking. But I think if I didn't go that direction, I mean, in some ways I like the writing part of science. Not all scientists do the writing,
Yeah.
but I think maybe something writing
Writing?
would have been an alternate career.
Like fiction or nonfiction, you think?
Nonfiction sounds like the safer.
Yes. Right. Richard, how about you?
So I thought for a while whether I should do veterinary medicine. I thought more like the in the field, and I think that would be my alternative.
Animals. Okay.
Yes.
How about you, Andrew?
Yeah, I really hadn't thought about this much, so I'm glad I got to go last. At least I had a few seconds to think about it. And I guess when I was younger, especially, I used to really like to argue a lot. So perhaps law would have worked out.
Yeah. Or politics.
Yeah.
Okay. All right. This one is easier, Priya. RNA or DNA?
RNA, all the way.
Chris?
I mean, I think I'm an RNA person.
RNA?
Oh, RNA. It's horrible when viruses have more than 8 open reading frames.
Well, I'm a host guy, so I have to say DNA.
Okay. There you go. All right. That's our Georgia State TWiV. And you can find all the TWiVs at microbe.tv/TWiV. If you listen on your phone or tablet on a podcast player, please subscribe. You get every episode. It's free, and we know how many people are listening. That's important for us to try and support the show. And if you'd like to help us financially, you can go to microbe.tv/contribute. We have ways that you could contribute as little as $1 a month at PayPal or Patreon. As always, questions and comments, twiv@microbe.tv. My guests today from Georgia State have been Chris Basler. Thank you so much.
Thank you.
Appreciate your time. Priya Luthra, thank you.
Thank you very much.
And thanks for inviting me and being persistent to make sure.
Yeah, I'm
came.
sure that all worked out, and we all are here.
And I was here to visit Georgia Tech, so you asked me to stay, and I think it was really interesting to do so because, frankly, there's no virology over there. I didn't find any, so I did a TWiV at Georgia Tech, which was very good, but here we have virology. Richard Plemper, thank you so much
Thank you.
for joining us, and Andrew Gewurz, thank you.
Thank you. My pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode was recorded, edited, and posted by Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.